Hello everyone, my name is Dr. Saoirse Tracy and I'm in the School of Agriculture and Food Science, and thank you very much for the invitation to speak today at the Research Day. So I understand one of the main aims of the Research Day is for researchers within the Institute to introduce themselves. So I've given you just a couple of slides of my background and who I am, but the main thing I want to talk to you about today is a technique I use called X-ray computed tomography, and I'm going to give you some examples of the applications of this technique across some food and health research areas at UCD. So as one of the aims was just an introduction today, I've just given you some details there. I'm an assistant professor in plant and soil science in the School of Agriculture and Food Science. My main research interests are root to soil interactions. I'm interested in the shape of roots and how they interact with soil to really help some of those key root functions like taking up water, taking up nutrients, anchorage, but also the health of the soil. So we know roots have a key part to play in the rhizosphere. Maybe how can we manage our agricultural production systems more sustainably to kind of maximize the benefits that roots would have to soil health. And I've used the X-ray CT technique as well. So my details are just there, and you can find me on Twitter as well. This slide, really quickly, just to give you some background, is my research sphere. So I've been using the technique of X-ray CT since 2008. I did my PhD at the University of Nottingham, where we looked at the response of root system architecture to soil compaction. One of the key advantages of this technique I'm going to tell you more about is that it's non-destructive. So we can look at roots in soil in 3 dimensions non-destructively. Before the advent of this technique, this wasn't possible. We had to separate the roots from the soil, so we'd lose a lot of this information. I then went on to do a postdoc in collaboration with the University of Southampton, where we still use the Xray CT technique, but we started to ask more powerful questions and use those actual geometries to make new mathematical models on how plant roots take up water and how water moves through soil. And I've been at UCD since 2015, so setting up my own lab, researching soil health, rhizosphere processes, and root interactions. So I've never really left the rhizosphere, and I don't want to, because it's a frontier for research and there's still loads of questions we need to ask. And I think they're very pertinent with some of the challenges we're facing in the coming years. So this technique of X-ray computed tomography, it could be new to some people, but if you've heard of CAT scanning in hospitals, it's exactly the same technique. So it's been around for decades, but now there are machines built purposely for research purposes. And these are the 2 types of machines we have at UCD. This is an example of what you would see if you looked inside a scanner that was built purely for research purposes. So in a hospital, the person would lie still and the X-ray gun and the detector would move around 360. In these machines, it's the sample that actually rotates. So here we've got the X-ray gun, the kind of yellow tube there, the manipulator stage. This is what rotates 360 degrees. You can see there's a glass rod with a small soil aggregate attached. The X-rays are fired from the X-ray gun. They pass through the sample here and they hit the detector. So this black panel is a key piece of the machine. This is where all the image information is collected. So this is the soil aggregate that we've got sitting on the glass rod here. And X-rays are taken at multiple angles as the sample's freely rotating 360 degrees. And this is what gives us essentially a 3D X-ray. So unlike the kind of classic 2D radiograph, we can build up all of these projection images, we call them, and reconstruct them into a volume. The key limitation to this technique would probably be sample size. So the smaller the sample, the higher the resolution. And these are the 2 X-ray CT scanners we've got at UCD. So through 2 SFI infrastructure grants, I was able to buy both the scanners. So we have the Vitamex. This was and they're both housed in the Rosemount Experimental Research Station, which is on the Belfield campus. And this is a larger scanner, a Vitamex. It can fit quite large samples. So maybe it says 30, but probably really more like 20 centimeter diameter maximum. and we can scan multiple scans, so we can have quite tall cores. So we've scanned, say, sediment cores or peat cores that are over half a metre long, and we can take multiple scans, but we can still get high resolution images. But we also have the Nanotom M, which is one of the highest resolution scanners available globally. Theoretically, it can get down to 0.3 of a micron, but then we're talking really, really small samples like a sand grain or a tiny piece of eggshell. So this is the scanner we use for the really high resolution where we've got small samples, but we want to get really good detail. And they say this scanner, well, this obviously is selling pitch, but this is what the companies say, that it's as close to a synchrotron image that you can get, but without any of the hassle of getting accessibility to a synchrotron. And this scanner, we take slightly smaller samples, but we could still take, say, 7 centimeters by 15 centimeters. So still quite reasonable sample sizes. And they're both available to use by anybody. It can be industry, academic research, from anywhere in the world. So if you want to access one of the scanners, just please drop me an email and I'd happily set up that collaboration. I know as well, we probably all would class ourselves as interdisciplinary researchers now. And it's something we're always increasingly asked to do. In my field, I think it's quite easy to do, but it's something I'd like to encourage more, and just be mindful of the tools and the techniques that are out there. So in this area, we've used applications from engineering. Advances in computer science has really helped this field. So especially advances in graphics cards has really helped us render the 3D animations. This is a daffodil bulb in some soil. Obviously as well, the scanners were originally developed in the medical field, and we're just adopting them in the agricultural sciences, and then mathematics as well. I think if we can take the best tools and the best techniques from several sectors, then we're really going to have really powerful research and answer some of those key research questions. So that's something that I think the Institute has been really helpful with, in kind of maybe matchmaking or setting up people from different disciplines. And the next slide will show as well how, since being UCD, the scanner's been used from lots of different schools, and just I want to give you some examples now of the different research areas the scanners have been used in so far. So the mainstay would be roots and soil. So we do a lot of scanning of roots and soil, because obviously that's my research area. We do root phenotyping, so answering basic fundamental questions on what do the roots look like in different varieties, what happens with the roots in different soil structures or maybe different tillage practices. We're currently developing a technique to look at parenchyma, so air spaces in roots that develop in waterlogged soils. And recently there's been an influx of lots of researchers working on root microbe interactions using the CT scanner. This could be mycorrhizal fungi, endophytes, nematodes. So we're answering lots of questions there. That image there is actually of a clover root in some soil, and we were looking at the effect of soil compaction on nodule formation in clovers. So we scan soil and plants from, say, pasture or tillage or natural systems. So anything roots and soil I'm interested in. We're doing a lot of scanning of above ground parts of the plants as well. So this technique can be very good for looking at seeds. We've been looking at hemp flowers recently. We've been trying to investigate fungi on the surface of leaves, looking at some plant pathology questions, different varieties, the influence of climate change. So we can scan roots, but we can also scan shoots. Anything basically you want to get a non-destructive view of an internal structure, we can use it for. In the food area, that image there is of a piece of cheese. So we were looking at calcium deposits in the cheese. We've scanned biscuits to look at the porosity of the biscuit, which is always a technique I like using, because I use the same technique, but for looking at porosity in soil. We've looked at, and hoping to do more, look at cellular structure of different foodstuffs. But I think in the food area, where the technique is used the most is maybe with companies trying to look at if they can adapt or tweak a recipe. Does that enhance the shelf life, or just looking at answering those fundamental questions. The engineering researchers use the technique a lot. So iForm have been doing a lot of scanning of 3D printing. We've scanned medical devices, we've scanned drugs. Metrology is just the discipline of measurement. So this is a really key area in engineering where we want really precise, accurate measurements of structures. So because this technique is such high resolution, we can be more accurate in terms of how small something we can measure is in terms of the resolution. So we've done a lot of scanning for researchers in engineering. And recently we've done a lot more scanning for archaeology. So we've scanned bones, that's a human inner ear bone. And with archaeological samples, obviously the samples can be quite precious, they can't be replaced easily. So increasingly archaeology is an area that's using a lot of X-ray CT scanning to keep a kind of permanent volume of that sample. So if they have to destroy it for other techniques, for say getting the DNA out, for example, they have to destroy the sample, but if they do a CT scan first, they then got it to archive. So future researchers could use and investigate the sample. So we've scanned fossils. We're hoping to scan some cattle molars to look at different diets. So there's lots of applications we can use, and this isn't exhaustive as well. We've scanned many other samples as well. So if you think there's a use of the X-ray CT to get this non-destructive 3D image, then yeah, please, like I said, drop me an email, which is just there. And thank you very much for the time today, and I look forward to answering questions.